It's a new class and you are well acquainted with this topic, white blood corpuscles, white blood cells or leukocytes. Leukocytes are one of the blood cells that too synthesized in the bone marrow, but lymphocytes can be generated other than this site. They can be generated in lymph nodes also. Okay, so I'll discuss. This is unlike erythrocytes. Mature erythrocytes do not have nucleus. Okay, but here we will be getting nucleus. So what we will do? We'll study these things. Why they are called white blood cells. When we centrifuge blood with anticoagulants, adding anticoagulants, when we centrifuge, we know how many layers we get? 2, plasma and cellular part. This is mainly RBC. In between, there is a coat. We call it buffy coat. And these white blood cells are present in the buffy coat, and this layer is white. That is why the term is given white blood cells. You will find all the cells here. In addition to white blood cells, it may contain what? So these are normal constituents. Other than this, abnormal cells. In diseased conditions, we produce abnormal cells. So they will also be present in this layer. So if we want to study whether the blood is having abnormal cells, so what we will do? We will take the buffy coat. We will take the buffy coat, and with this buffy coat we will make a smear on a slide. Then we will stain the smear. Then it will be studied under microscope. To look means blood film. Film. Film means smear. Okay. There may be some abnormal cells. It may be cancerous cell. It may be abnormal white blood cells. So, if there is any abnormality in cells circulating through the blood, this can be found in buffy coat. So, it is very important too. These are nucleated. We know that these are nucleated, and mainly their function in defense mechanism. Okay. They have other functions also. We will discuss. They destroy the invading organisms and destroy abnormal cells like cancer cells. What is the normal values of the WBCs? 4,000 in some books it is written at 4,500 and up to 11,000 per microliter or per cubic centimeter or millimeter. Millimeter, right. Don't be confused. Okay? At birth their number is much more. This is the gradually when they attain 3 to 4 years, they gradually starts coming to normal level. Neutrophils, these are called differential count. See, when a patient is advised for blood test, so it is written TC, DC, BSR, hemoglobin. So DC stands for differential count, because we know WBCs are of different types. WBCs are of different types. So differential counts means white blood cells are of different types. What are these? Neutrophils, Basophils, Eosinophils, monocyte, lymphocytes. So first, all the WBCs will be classified according to the presence of granules within their cytoplasm. Then we divide them, all the white blood cells, into two. Those who are having granules in their cytoplasm and those who are not having. So it means granulocytes and agranulocytes. Granulocytes, when these granulocytes are stained with some stain, say a Leishman stain, okay, we have to differentiate the cytoplasm from the nucleus. So nucleus will get one type of color, and there, because the content, chemical composition of nucleus and the cytoplasm is different. So based on that, mainly the nucleus is acidic. So they will take basic stain. But the cytoplasmic granules may take, since they are basic, mainly the proteins, so they will take the opposite acidic stain. So in this way, Leishman stain is having both. So they can stain the acidic, the cytoplasmic granules as well as the nucleus. So both will be appearing or giving different color. So it will be possible for us to diagnose or to identify what are the cells actually, in which group it belongs, whether it is granulated or agranulated. Again, depending on the granules, color and size of the granules, we classify them. Depending on the nucleus they are possessing, we classify them. So based on what? Based on what we further classify granulocytes? The type of granules, their affinity for the stain, okay, and the nucleus. We classify them into three groups. One is neutrophil, another is basophil. Neutrophils means it will take neutral stains. I mean, they will take acidic as well, both basic stains. And Eosinophils will only take acidic stain. Whereas the other cell is basophil. Basophil will take basic stain. So in that way, we classify granulocytes into neutrophils, eosinophils and basophils. And the other two, lymphocytes and monocytes, they don't have any granules. They don't possess any granules in their cytoplasm. So they are agranulocytes. Agranule means nil, no granule. Agranulocytes. Okay, they don't have. So among these lymphocytes, origin of lymphocyte is different from monocyte, though both are agranulocyte. Origin is different. We will study later. What is the percentage differential count? See, right hand side, these values are written in differential count. It is given. So most of the cells are neutrophil, and in disease condition, in some disease condition it will be reduced, and some disease condition it will be increased. When bacteria or we get infected by any foreign organism, so it happens, it has to fight with this organism, so its level in the blood is increased because of increased formation of, there will be increased formation of WBCs, means the process is called leukopoiesis. Like erythropoiesis, leukopoiesis means production of WBCs from bone marrow. So leukopoiesis will be stimulated when bacteria or toxins will be invading in our body. Whenever any infection comes in my skin, immediately they will come, they will reach to the site. How they come to the site, how they fight, what is the mechanism that I will discuss in detail. So what is the percentage? Second highest is lymphocytes. Second highest, what is the percent? 20 to 40%, and neutrophil is 50 to 70%, and the minimum, which is the lowest, basophil. If you look for 100 WBCs, you will get one. That's why it is 1%. In your histology class, you will be looking for basophil, basophil, basophil. Sir said basophil looks like this, but I am not getting any. Or you will find one cell which is not actually basophil. Sir will say, "Are, what is this here? It is not basophil." Okay. So the reason is this: its number is less. Its number is very less. So you have to move around the slide under the microscope where I can find. Let's see. You have to look for it. Never let monkeys eat banana. Okay? So if you remember this 60, 36, 3, 1, that means this is their parasitis. Okay? That's why it is given. Now, we will discover variations in WBC count. We said sometimes it is increased, sometimes it is decreased. So, we use two terms. One is leukocytosis and one is leukopenia. Penia means low. Penia means low, lower than normal. Cytosis means increased production beyond the normal level, normal range. So cytosis, remember this, leukocytosis. Another common word is there, pancytopenia. Pancytopenia. Meaning what? In some diseases of bone marrow, because bone marrow is the responsible site for producing all type of blood cells, excepting lymphocytes. So, in such cases, all the cell varieties will be reduced. Penia means all the pan, means whole. All the blood cells will be reduced. If there is any disorder in bone marrow, production of all variety of WBC will be, and the term is pancytopenia.
So if there is any defect in bone marrow, what will happen to the erythrocytes? They will also decrease.
Yeah, of course. There will be anemic. If the patient will be anemic also. Why there is anemia is? Why there is anemia? So, I will study hemoglobin, I will study erythrocyte count, both are less. PCB is less. Then why it is? What is the cause? I will check the iron. अरे blood में तो iron है ज़्यादा. Normally है. Then what are the reasons? Then okay, you have to go for bone marrow analysis, bone marrow biopsy. So you have to take bone marrow either from this bone or from the hip. There are so many sites. So from that site you have to take some, aspirate some marrow. Then you have to study under microscope, and we will find that there are some myeloid, abnormal myeloid cells or cancerous cells, or you will be able to diagnose whether there is leukemia. So in this way, abnormality of in bone marrow can be studied. The patient will be sent to the hematologists. They will say what is actually happening in bone marrow. So leukocytosis can happen physiologically. So these are some, not pathologically. There are some physiological conditions. In newborn baby there will be more, and gradually it will be coming down to normal. When you start exercising, then it may increase. When you are in stress, any type of stress, then its level will be rising. Its level will be rising. Stress means there will be liberation of ACTase. Whenever any form of stress, there will be liberation of ACTase. So this ACTase will cause secretion of cortical hormones, and these cortical hormones will increase the erythropoiesis. So automatically, not only erythropoiesis, in leukopoiesis also, as a result, there will be during stress, there will be increased leukopoiesis. Pregnancy is the obvious reason. So in pregnancy it also rises. After food intake, temporarily it also rises. Then again it comes down. Actually, their life is very short. When they are produced, they will be entering into the blood. Few hours they will be entering into the blood. 4 or 5 hours only. Then where they will go? Where they will go? Where they will go? Can you suggest where they will go? But their life is not ended. They formed in the bone marrow, liberated in the blood, but here it will not stay long. It will pass out from the blood vessel, will be entering into the tissues. And to protect your tissues from bacteria, invaders. They will take the first line of defense to protect you. So they will fight in the tissues. So they are not permanently, they are not staying. And there in the tissues, they will be surviving for 4 or 5 days. Thereafter they will die. New cells will be coming. And some cells, it is so unique. This is the blood vessels. Some WBCs remain adhered to the inner wall of endothelium, inner surface of the endothelium. When you require, when there is infection, immediately they detach from the wall and either it will enter into the, start circulating and will be coming out from the vessels to the infected area to fight the invaders. Okay? Can you follow my class? Sure. Or any problem is there? You. Yes. No problem. Then I will be asking questions. So you have to be very attentive what I am telling. Immediately I may ask you questions, so you have to answer. So it is for applicable for others also. So keep your concentration. And there are some pathological conditions in which WBCs are raised. There will be leukocytosis or leukopenia. We are talking about leukocytosis, the conditions in which level is raised. One, we discussed about physiological conditions. Now I am discussing about the pathological conditions under which there will be leukocytosis. What are these? Acute bacterial infections, acute hemorrhage, burns, tuberculosis, like many other diseases. In that case, we will case pathological increase in WBC count, or that is, we call it leukocytosis. Leukopenia is the just opposite of leukocytosis. Means decreased production, decreased quantity, decreased count in blood. So when there is decreased in quantity, then my body is in trouble. Who will fight for me? So I will be very prone to different diseases. So anytime any organisms can attack me. Leukemia is a cancerous condition of blood in which the TLC, means total leukocyte count, is more than 50,000. Normally it is up to 11,000. So now it is 50,000 per in case of leukemia. That too, these are abnormal. That too, these are not normal. That too, these are abnormal. Associated with presence of immature WBCs in the peripheral smear. So we divided different type of WBCs into granulocytes, agranulocytes. Granulocytes are neutrophil, eosinophil, basophil, and at the bottom main functions are given. And at right hand side there are agranulocytes, lymphocytes, monocytes. How they look, it is also been shown. And from lymphocytes, there are three other varieties of lymphocytes are there: T lymphocyte, B lymphocyte, and killer cell, natural killer cells. They fight against cancerous cells. So that's why lymphocytes is very important. And this is various stages of erythropoiesis. I mean leukopoiesis. See, this is the stem cells. This is the stem cells. This picture is taken from your book, G.K. Pal, hopefully. Is it G.K. Pal? AKG. Okay, it is AKG. So you see, this is one series, this is one series, this is one series. Okay, these are basophils. See the granules. And this is neutrophils. These neutrophil granules are very fine, sand-like granules. Here the granules are coarse, and they take basic stain. And nucleus is also there different. Here the neutrophils, these are the basic steps. You don't need to study in detail, because there will be allotted either maximum 4 or 5 marks. So what are the steps? You remember the steps. Okay, these are the steps. And this is given in G.K. Pal. See, neutrophil. Pluripotent stem cells, main stem cells. Now they are directed, they will be differentiated depending on the demand of the body. They will differentiate into different. Suppose I need neutrophil. What will do? They will divide into myeloid series. Another is lymphoid series. Lymphoid series will provide lymphocytes. And lymphocyte, 2 varieties of lymphocyte. And the myeloid series, they will provide erythrocytes, granulocytes, and monocytes. This series will make this basophil, eosinophil, neutrophil, and erythrocytes. And this series, from pluripotent stem cells, these progenitor cells are produced. From this progenitor, this will decide whether to make T cells or whether to make B cells. Okay, so now we want neutrophil. There is invasion of bacteria. So why there should be more erythrocytes? So what will there? This line will be activated. This line will be activated, and ultimately it will produce neutrophil. Clear? Okay? You have to remember these steps. Once again. Some bacteria invaded your body, so you need to fight. So the stem cells will recognize it, because there are some factors, various factors are there. They will cause disease needed. At the side, IL-1, IL-3, IL-6, colony forming stimulating factors. There erythropoietin is there. Here erythropoietin will not come into action, because erythropoietin's function is only to increase erythropoiesis. So it has got no role here. The others are having role. Other factors are having role, as I have discussed in erythropoiesis. Okay?
Leukemia, does it happen in autoimmune conditions?
In?
Autoimmune conditions, autoimmune diseases.
So it will sense I need neutrophil. So accordingly it can generate any type of, these are hemopoietic cells, any type of blood cells it can produce. When the body needs neutrophil only, suppose, then what will do? So it will produce this myeloid series cells, because all this comes under myeloid series. So now this myeloid series cells will be committed. So we will now commit to produce either granulocytes or erythrocytes or megakaryocytes or monocytes. So what is written there? CFU GEMM. G for granulocytes, E for erythrocytes, M for megakaryocyte, and another M for monocytes. See, now they are differentiated into now these are differentiated into E. E means erythrocyte, so they will produce erythrocyte. Now this is again another series is the CFU megakaryocyte, okay, and GM means granulocytes. Okay, so we want neutrophil. So from here neutrophil will come. Again these will be divided into granulocytes and monocytes. Now they will differentiated into granulocyte, neutrophil. This is, it will be, instead of G it will be E. Okay, and it is not G, it is E, and CFU eosinophil, and it is basophil. So I need neutrophil. So root is this, then this, then this, then this. This is the root. Understood? If I need eosinophils, my root will be this. These are the steps. So we will be writing these are the steps. Understood? Clear? Not clear? Say yes. Huh?
Sir, clear.
Clear, okay. How the shape, see, when they develop, so how these are changed. See the condition of the nucleus. Huh? The last stage is neutrophil, mature neutrophil. Before that, it is neutrophil band. We wear band, no? So nucleus is band shape. Now we are coming in detail. So these are the steps for neutrophil production. Now we'll discuss neutrophil. Now our point of discussion is only neutrophil, not others. That we will be discussed in other days. Let's see what it does. Neutrophils, those who are, those are band shaped, no? These are very infant. These are very young, newly formed. When they grow, no, then this band shaped nucleus will be lobulated. First, but they are interconnected. First, they will form bilobe neutrophil. When they will be maturing further, aging further, there will be trilobe neutrophil. Again, there may be tetralobe, 4-lobe neutrophil, 5-lobe neutrophil, 6-lobe neutrophil. Then they will be dying. These are old, senile. So 6, if you are having lot of 6-lobe neutrophils, means new cells are not producing in your body. But if you find lot of band-shaped lobe 1 or lobe 2, means young is more, means body has been invaded by foreign organism. For which it is now started producing. It has to fight, na? Young are coming to fight with those. So they will be maturing gradually. So presence of So what we will do, we can another index from this, whether it is monolobe, I mean band shape, means one nucleus and two nuclei interconnected, means bilobe, trilobe, tetralobe, pentalobes, hexalobes. So let's see. Hello, let's see. What is innate index? You are given 100 numbers of, you have studied 100 numbers of neutrophil. And within this 100 numbers of neutrophil, there will be band, there will be bilobe, there will be trilobe, there will be tetralobe, there will be pentalobe, there will be hexalobe. And see what is the percentage of each one out of 100. If you find band shape it is 3, then it is 3%. If it is 12, it is 12%. So in this part, then you, if you plot a graph, so at the x-axis, monolobe, bilobe, trilobe, tetralobe, pentalobe, and this y-axis will indicate numbers. Out of 100, what is the number for one lobe, what is the number for two lobe, what is the number for three lobe, this way. Then we will connect this point, this point, this point, this point, and we will get a curve like this. If it is a bell-shaped curve, ding-dong bell-shaped curve, then it is normal. The rate at which it is producing, the rate at which it is dying are same. And at the center means their number is more, they are middle digit, not like me, I am old digit person. Okay. One means band shaped, two lobed, three lobed, four lobed, five lobed, 6. If you find 7 also you can get sometime. And here is the percent. 10, 20, 30, 40, 50, 60, 70. So there is only how many neutrophils? 100. So these 100 will be distributed among others. Okay. So now if you find the curve like this, bell shape, then it is normal. If the curve is like this, new generations are more, old generations are less. That means your body may be infected. Because there is some purpose so coming in higher amount. Somehow your bone marrow has been stimulated to produce more through neutrophils. It has to fight. So in body infection, recent infection, you will get this type of innate index. This is called innate index. And if you get curve like this, you are getting radiation. So there will be less production. Now only the old neutrophils are staying, so their number will be more. New will be less. So curve will be shaped like this. Understood? It is now clear to you what is innate index? Very good. What is this? Trilobed. It is band. It is bi. This is the link. There should be link. See, there is a link. There is the link. This is the link. And it is 1, 2, 3, 4. So in your practical class, You will be staining the blood smear, then you will be, after staining, you will be observing under microscope, you will find lot of neutrophils. The RAs are told this is tetralobe, this is pentalobe, this is bilobe. In that way, you will be able to and show us, sir, I got one bilobe, I got one trilobe. Okay? So their morphology. As I said, they have granules, the fine granules, and they may take light pink or light bluish color. Blue to pink coloration, colored granules you may find, but they are very fine. And granules are fine sand-like and take acidic and basic stain, I said, so that's why we call it neutrophils. Contains variety of enzymes. We'll see lot of enzymes. What are these enzymes? Lot of enzymes they possess, and they are fighting all for the purpose of fighting with the invading organisms or the toxins. Okay, they will help. We know this WCs can move like amoeba. They can form pseudopod. So they will produce like pseudopod, and with this help of this pseudopod, they will change their shape and will be elongated, thin and elongated, and we know they don't remain within the blood vessel. They come out into the tissues and they travel within the tissues. So what they do? This is capillary, blood is flowing, so they are having lymphocytes, are neutrophils, they are having neutrophils, endothelial cells. So in between the endothelial cells, there are some space. These cells, which when they are to come out of the blood vessels, they produce pseudopodia, they become elongated, and will enter through this space, and now there are the tissue cells. Okay, so they will come into the cell. Ultimately, they will reach to the infected area, maybe skin surface. So this way, and this procedure, this process is called what? Diapedesis. See, there are 4 varieties of granules in neutrophil. There are 4 varieties. One is primary granule, another is secondary granule, another type is tertiary granules, and secretory granules. So all these granules, see what the primary granules contains. These are the enzymes. Secondary, these are the enzymes. These are some proteins. Tertiary granules, enzyme. Gelatin is an enzyme. So all these things are required, the weapons. All these chemicals, all these proteins, all these are antigens, antibodies. They are weapons used by the military. Okay, different types of weapons we are having. So these granules are having different types of weapon, and they will use this weapon to fight the bacteria, to fight the invader, to fight the toxins. Okay. Because I have to finish, so I'll make it cut short. How they move? First, there is chemotaxis. It will smell. some foreigner have invaded. When they invade our body, they produce some chemicals. And we have detectives. We have detectives. They identify this is the thing. So they inform the neutrophils. How they inform? They produce, they are called complement system. We'll study complement system. Very complicated process. This complement system, lot of chemicals are involved. So these are liberated, or the cells which are infected, which were infected by the bacteria, they will produce some chemicals, and these chemicals stimulate the neutrophils. Actually, neutrophils get stimulated by those complement system. In that area, some invader has entered. So we have to go there. We are in blood, we have to go there. So this is called, getting this sensation from the infected area. It may be damaged cell, it may be the bacteria releases some toxins. It may be the toxins. So they are chemotaxis producing agents. Means we call it chemotaxins. So they will liberate chemotaxins, and when there is any chemotaxin in some area, immediately neutrophils are. There are different types of granules in the cytoplasm of neutrophils. We call these granules cytoplasmic granules, and they are of different types: primary, secondary, tertiary, secondary granules. What are the contents of primary granules? These are the contents. Secondary granules contain these. All these are weapons to kill the bacteria, to kill the invaders, to destroy the toxins. Honor count discussed. Now the neutrophils has got 4 different pools. During their generation, in bone marrow, they will be there. You will find there. If you study the bone marrow, you will find neutrophils are there. So these are marrow pool, and they remain in developmental stage. So different stages of development we have seen. So they will be there. We will get this in the marrow. So that's why this neutrophils we call developmental pool. Pools means, what is the pool? Car pool. There you will get numbers, they remain accumulated there. And then in circulation pool, see only 8 to 10 hours. In some book it is written 4 to 6 hours, 4 to 5 hours. So anyway, very short time. And after that, when they are released from bone marrow into the circulation, so there it will remain for a very short period of time. Thereafter what happens? They remain marginated. Means what? Means blood vessels, capillaries, these neutrophils remain marginalized on the epithelial line. So they are stocked there. Whenever I require, this will be disorganized, means they will be freed. Now either they will remain within the circulation. Suppose circulatory amount has reduced. So immediately this will be disorganized. They will be detached from the wall, demarginated, and will come into the circulation. But if they are needed in the infected area, to the place where foreigners has come. So from there they come out of the blood vessels and go to the area of infection. So that is the importance of marginated pool. So they are ready for the fight. Whenever I required, we will go to the site of occurrence. Next is tissue pool. Here they will remain for 4 to 6 days, 7 days maximum. After entering into the tissue space, so they will be staying there for this much time. Huge number of neutrophil is eliminated daily because their life will be finished, no? So they will be eliminated and there will be macrophage will take up them. And they will take, then it will be exocytosis, endocytosis. You know, what is exocytosis? What is endocytosis? Movement.
Endocytosis inside and exocytosis outside. Movement.
Something you are to taking in within the
Cells. Cytoplasm.
cytoplasm through plasma membrane. So that is endocytosis. You are eliminating something. Exocytosis. Because this is not diffusion. Then you will say this is like, what is the difference between diffusion and this? The molecules are big. See, I need a protein. I need somatotrophin, growth hormone. Why? For my growth. So it will be acting, it is produced in pituitary. Here it will not be acting. So it has to travel to the organs. Okay, where growth will be there. Okay, then how it will go there? Growth hormone is a protein, big molecule. It cannot pass the membrane for where it is synthesized, the cell from the cell to the blood by simple diffusion because protein molecules are big. So what is happening actually? Nucleus produces DNA produces mRNA. This mRNA will travel through the endoplasmic reticulum, rough endoplasmic reticulum. What is the function of smooth endoplasmic reticulum? Lipid, not secretion, synthesis. Secretion is one thing, secretion is after it is synthesized. And rough endoplasmic reticulum, protein. So our insulin will be produced there. In our beta cells, they are producing insulin. These are protein. And they will be produced in the endoplasmic reticulum. There are ribosomes, 40S, 60S, different type of ribosomes are there. So this mRNA while passing through this, so it will dictate how to form the peptide chain. What would be the sequence of amino acids in the chain? Who will be first, who will be the second, who will be third? And if third is supposed to be valine, it is not there. Then, okay, this one is available, valine is not required, it is there, so you put it. Now it is not that very protein. Now it is something else. Or it is an abnormal one. If you replace one of the amino acid by another one, then it will lose its property, it will lose its function. It is so unique. Okay. So after their peptide chain is formed, then they will be packaged like factory. One thing is produced, now they will be polished and will be packaged. Where it is packaged? In Golgi bodies. This will be packaged. So Golgi bodies are elongated. So here will be packaged. So one part will come out. With this is what? Vesicle. Containing this protein. And this protein is required by other tissues. Then how it will be going out? So then this packet will go to the cell membrane, and this cell membrane will by mechanism of an exocytosis will break this and liberate the contents into the extracellular fluid. Now nowadays they will be transferred into the blood capillaries. From the blood capillaries it will go to the target organs. Understood? So this is the process. So by the process of when the cells are damaged, either in our body when it is infected, pus is formed, white colored pus is formed. This is what? Not only WBCs.
Many other things.
WBCs are there, RBC, some blood will be, that is RBC, dead RBC will be there, dead tissue will be there. Okay, so, and they are coming out from a skin after some days. Okay, it will create pain and ultimately it will come spontaneously. Okay, or by putting incision we take it out, the pus. So pus is formed because of this fighting. And the fighter, macrophage, neutrophil, they will also die. Army also, during fighting, army also die, no? So they will also die. So how they will be eliminated? They will be taken up by the macrophages, they will be particled, and they will be exocytosed from the cell, from the macrophages, and will be liberated. They will be again coming into the blood, and through our kidney, circulatory system, excretory system, they will be coming out. Okay, so this is the process. Now the functions of neutrophil. What is the first function? This is actively phagocytic. This is actively phagocytosis. So what will do? What is phagocytosis? Suppose this is the invader. So the cell will produce pseudopod, will engulf this and will bring it within the cell. Now this will be combined with lysosome. Lysosome contains various enzymes, proteolytic enzymes, different types of enzymes. So they will digest this. They will first break down the cell wall. Within the cell wall there is cell membrane. Okay, so some enzymes will destroy the cell wall. Then some parties will destroy, some enzymes will destroy the cell membrane. Then it will destroy the nucleus. Okay, some enzymes are there, active enzymes are there, they iron containing enzymes. Some will split off this iron. So this active ingredient within the toxin, or this will be eliminated, broken. So in this way there are various mechanisms. Also we will find. Also, have you heard about antioxidants? We take antioxidants. Why? What actually antioxidants do?
Remove toxins from the body.
Some oxidants are produced in our body. They are very deleterious. They destroy our cells. Okay, so we prevent this destruction by taking antioxidants. We will see. These granules contains oxidants, make oxidants to kill the bacteria, to kill the bacteria, to kill the organism. How these oxidants are formed? Can you name some oxidants? O2 negative. O2 oxygen negative. negatively charged O2. These are oxidant. Hydrogen peroxide. Hydrogen peroxide. Hydroxyl group, OH negative. HOCl. These are oxidants, harmful for the body. So, with these weapons, with these oxidants, they destroy the bacteria. And when firing is there, innocent people also die, no? So, around this, some tissues are there, so they will also die. That's why they sacrifice and pus is formed. Because of the toxins liberated by the bacteria, foreign bodies, they produce some toxins. Sensing these toxins, actually, our neutrophils, they can sense these toxins, so they come out of the vessels, go to the affected area and fight. Neutrophils, second function was what? They are considered as the first line of defense. Okay? Against acute bacterial infections. They contain another. When we are infected, we get fever. Why? Because these neutrophils contain some pyrogens. Pyrogens are fever producing. When they travel through when you inject some pyrogens or you contact some pyrogens, say, sometimes in saline, saline, it also contains pyrogen. When saline is given in some cases, so immediately there is temperature. Okay, because it also may contain pyrogen. Okay, this pyrogen, when travels through blood to our hypothalamus, and it suppresses the activity of the temperature regulating centers. We have temperature regulating center. One, for if temperature is raised, the nervous system will make some mechanisms, processes it will start, so that heat is liberated from the body. This is like excess sweating, reducing production of energy, breakdown of energy liberates it, no? So I will minimize the catabolism. I will minimize the secretion of thyroxine. It increases metabolism, increases heat production, increases BMR. BMR is what? Your BMR is high, means you are liberating more energy, more heat to the environment. So it will manage. If there is excess heat production, there will be sweating. You will feel hot, so you will switch on the fan, you will switch on the AC. And your body, you will open the dress. You will open the surface, you will open the surface area of your skin. What for? Because when surface area is open, it will eliminate heat in different processes, physical processes. What are these? These are, okay, he is having temperature higher than me. If I hold him for some time, what will happen? My hand will be warm. So why his body temperature is transferred to my hand?
Because of temperature difference.
Obviously, but what is the mechanism? Sunlight. You went to the field. Sunlight is there. So you will feel hot now. So how this hotness, heat from sun came to your body? What is the process? What is the process? So there are different physical processes. Convection, conduction, convection, conduction, evaporation. Conduction. Example, you are cooking with a spoon. So other end is getting hot. So heat from here is conducted through the handle to my hand. So this is the process of conduction. And convection, we are eliminating heat. So this is coming into air. From air is becoming hot. Then my temperature, my body also will, if it is lower than the surrounding temperature, so I will get heat from the temperature. It is convection. Through air. Transfer of, exchange of heat through air, through a media. So this is convection. Another is irradiation, radiation. The sunlight is coming to earth, to our body through radiation. So what are the physical processes? Radiation, conduction, convection. Another process of removal of heat from our body is evaporation. We said, when there is hot, we liberate sweat. Now, this sweat is evaporating and the sweat is liquid and it is evaporated. So, it will be taking some heat from me and this is the latent heat. So, through this latent heat, we are also losing heat. So, these are the mechanisms. Again, if it feels, if the center feels there is body temperature is low, so this nervous system will try to increase my body temperature. How? What are the mechanism? It will stop or minimize the circulation of blood through the skin. Cutaneous circulation will be minimized so that their heat is not coming to the surface, not going out by the physical processes. So heat will remain within the core of my body. So this is one mechanism. There will not be sweat secretion, and if it is too hot, too cold, then what we do? We shiver. We shiver. There is muscle tremor. We start shivering. Everybody have faced this. We after taking bath in cold water during winter season, we face it. Okay. Why do we shiver? We shiver. What for we shiver? Because shivering means muscle contraction. Breakdown of ATP means liberation of energy, liberation of heat. So, by that way, we can increase our heat. We can increase the secretion of thyroxine. So, there will be increased heat production. Okay, so these are the mechanism our hypothalamus made. Temperature regulating center. This is the temperature regulating center, and it understands whether my body temperature is high or low by our thermoreceptors. We have external thermoreceptors, we have internal thermoreceptors. With this, we sense whether my body temperature is at 30, what is the normal body temperature? At 30, 37, 38, 98 degree Fahrenheit. I said, whenever you tell me the value, so who will say it is centigrade or Fahrenheit? You are supposed to tell, no? Okay. When pyrogen is there, when this pyrogen will be passing through my hypothalamus, it will disturb my regulating centers. So now it is unable to control the body temperature. It is because of the pyrogen. So whenever there is infection, so the neutrophils will be going there, they will liberate pyrogen, and this will pass through the blood and will affect the thermoregulatory centers of hypothalamus. Now what will happen? There will be increased production of temperature. There will be increased production. This is what fever we call. Another term, febrile. Fever, you will be writing the patient is in febrile state. Not you will not write fever.
So pyrexia.
Huh?
Pyrexia is also fever. Pyrexia.
Pyrexia means fever. Pyrexia is also fever. Neutrophil, now one of the main function we see, phagocytosis. Now let us see how phagocytosis is done. What is the mechanism? So this phagocytosis mechanism has many steps. What are these? What are these? Number one, chemotaxis. Secondly, margination and pigmenting. I said no, they stick to the wall of the endothelial lining. So this is margination or pigmenting. Pavement what? Footpath. So along the wall, there is a footpath for neutrophils. Whenever we need smooth traveling, then what will you? Come out from the footpath and join us. Okay. Chemotaxis, margination and pigmenting is the second step. Then diapedesis. Diapedesis means they will be coming by forming pseudopods or by making them elongated. So they will be passing through the endothelial cells. They have some space in between. So through this space they will come out and will go to the affected area. This is called diapedesis. Okay. So diapedesis, after diapedesis, what is? Opsonization and adherence. This is food, but it is raw. Let's put some salt. Let's put some so that it becomes swadist, tasty. So opsonization means neutrophils will liberate. The neutrophils will liberate some chemicals which will cover the foreign body. And now the neutrophil becomes very much interested to engulf it. That is called opsonization. So this, where from these chemicals come? Come from the granules. It is having granules, and some granules are liberating those chemicals. And now it is attracted and easy. It has been encircled by this chemical. This is called opsonization. Because of opsonization, now the cell, neutrophil, now it is very easy to become attached to the foreign body. Foreign body cell membrane and its own cell membrane will come to each other. Now this will be adhered to each other. Understood? Okay. So this is opsonization and adherence. And then eating. And thereafter killing. Eating means taking in within the cell. Then killing. Now you face why you have entered in my body. Face the consequence. This is from your book. I think, which book? GK part, okay. Tissue injury, this is the epithelium, and this foreigner entered. This is the foreigner entered, and they release some toxins. These are chemotoxins. Now, whenever there is any chemotoxins here, our complement system sense it immediately, they react. And now, this neutrophils traveling through the blood vessels, they can sense it. Hari, something is produced. I can hear firing. Let's go to the battlefield. So, it is traveling, and this is the process of diapedesis. Now, it is coming, engulfing, and then it will start digesting. I said the complement system will be involved. So these are the complement system. C5a, C3, these are complement system. They and some other components, leukotriene B4 complement system. So they will be involved. Whenever this process, chemotoxins are liberated, immediately what happens? My bone marrow starts making new soldiers. New soldiers means new neutrophils. Diapedesis. The process by which neutrophils pass through the junctions between the capillary endothelial cells to reach the invader in the tissue is called diapedesis. What is diapedesis? It is written. When these neutrophils are activated, they are marginalized, and after that they will leave the capillary blood vessel by diapedesis, and this is called emigration. So this is the process of diapedesis. Then opsonization, to make it the thing tasty. What does it do? The process by which the bacteria are made tasty to the phagocyte is called the opsonization. Phagocyte and neutrophil are same. Okay. In this process, antigen, the foreign body. It may be bacteria, it may be toxin, whatever it is, it may be. So this process, antigen is coated by the opsonins. Opsonization, coating is there. So immunoglobulin IgG takes part in this. IgGs are antibody. So their role is very important. And attachment of the membrane of phagocyte to the membrane of the microbe is called adherence, as I said. So both the bacterial cell membrane, bacterial cell wall, and cell wall of the neutrophil, they will be attaching. After attachment, then will be the process of engulfing. So see, so this will gradually cover, coming and attaching. So opsonization, this is opsonization. Now, when it is opsonization starts, this is our neutrophil, it will now engulf this. It started engulfing. Now it is engulfed. Okay? Ingestion. The membrane of phagocyte extends projection from how the pseudopod like structure is formed, and thereby it is ingested, and then after ingesting it into the cytoplasm, there is also lysosome. There is lysosome, so it will attach with the lysosome. Lysosome will liberate its energy, its enzymes, so that the material now the foreign body has entered. Now whatever it is there, everything will be destroyed. It will be digested. Protein is there, this will be there, are elastase, so many protein digesting, carbohydrate digesting enzymes, different type of enzymes are there, they will make it digested. And it will eliminate amino acid, glucose, the basic components. Is it understandable? Bacteria. So phagocytosis, this is the neutrophil. Phagocytosis is done. Phagosome within the cytoplasm. Phagosome, now this will combine with lysosome, and now lysosome will liberate the enzymes and it will digest the bacteria, red colored bacteria, into pieces. Now the pieces will be exocytosed. Clear? Another process of the killing. Two processes there. Killing will involve two processes. One is non-oxidative process. Means this is an enzymatic process. There will say if it is made up of elastin, so elastase enzyme is there, this will break down. So this is the enzymatic process. And in non-oxidative mechanism, neutrophil granules contain variety of antibacterial chemicals such as degradative enzymes, proteases. Proteases are the enzymes that convert big size when you start digesting protein. Initially proteases come into action. Okay, then only peptides will be formed. Number of peptides will be formed. Then dipeptidase, tripeptidase, they will be coming into action and will produce. These are the very different. When we will be studying the digestion of protein, you will understand. So initial part is done by the protease. Lysozyme contains lysozyme. They hydrolyze the cell wall. Lactoferrin. I said they sequester the iron content that the bacteria contain. So it will be losing the activity. The bacteria will be losing the activity. Defensin, it kills bacteria by disrupting the outer membrane and breaking single strand DNA. If a bacteria loses DNA, then it is unable to multiply. So in this way, it kills. So by non-oxidative mechanism. Now the interesting part is oxidative mechanism. It will, the cell, that is the neutrophil, it will produce oxidants. It will produce oxidants. How these are produced? See, NADPH we have heard, there is hydrogen ion, there will be hydrogen ion within the cytoplasm, and because it's an acidic environment, inside is acidic environment, as a result, and when this process starts, our breathing is increased. Why? To supply, because at that time, oxygen demand by the neutrophils is increased. Okay, so it combines with this oxygen, hydrogen, and this will form NADP plus 2 hydrogen ion, and this is one of the oxidants. O2 negative, one of the major oxidant. Then this 2O2 and hydrogen ion in the next step, 2O2 negative plus hydrogen will produce hydrogen peroxide. This is also another oxidant. And some of the amount of hydrogen peroxide will again combine with oxygen to form OH negative plus oxygen. So whatever oxygen it produce, part of this oxygen is utilized in hydroxyl group formation and in hydrogen peroxide formation. So now what are the oxidants? This is one oxidant, this is one oxidant, and this is oxidants. And they are potent oxidating agents. So with these agents, they kill the bacterial bacteria. They have killed the foreigner. Okay. As a result, it is killed. You know, neutropenia means neutrophil count will be less than normal. What are the conditions? These are the conditions. Take a photograph of this. What are the conditions? What are the conditions in which there we will find neutropenia? So these are the conditions. Okay?